For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.
All right, looks like we are live. We got some good people in the live chat. I've been looking forward to this one all day. We've got Salvador Cordova back with us tonight. Protein Probabilities Part 2. If you have not yet seen Part 1 to this lecture series, please check it out. It is in the description box. So I've also got John Maddox with me here as well to interject questions here and there on the topic. So we're going to have a lot of fun. Also, guys, tonight around probably 2 EST, I'm going to be going live again to respond to good old Dr. Dan Stern Cardinal's latest rebuttal video. So looking forward to that as well. Make sure you are here. And right after this lecture, we'll probably keep it around an hour and a half, just so everybody can head over to Brother Sal's channel for an after show. So I'm looking forward to it. Enough from me, though. Let's hand it over to Sal. You guys are muted. John and Sal, if you guys want to speak, make sure you unmute yourself. And Sal, thank you for being with us here tonight again. I'm looking forward to it.
Well, thank you. Thank you. Thank you for everyone who's tuning in live and also through recording. God bless you all. So let's get right to it. Studying cellular biology and biochemistry can be tedious, so I'd like to offer some reasons to be motivated by this evening's topic on protein probabilities. So let me share my screen.
Okay, I'm going to share it right now. And before you go, Sal, let me just screen share. George Bond in the house with the super chat, $5. George, thank you so much. He says, because Sal laughs at my jokes. So, George, you are the team comedian. We always love your jokes. And yeah, John, he just posted the after show link to Sal's channel. Make sure you go over there, like, subscribe. We're going to have some fun. So let me, Sal, you want me to put your, I'll make this full layout. Let me see, full for your slideshow. There we go.
Oh, thank you. And George, I have a notebook on your jokes. Like, what kind of clothes do kangaroos wear? Jumpsuits, of course. Anyway.
Uh-oh, George, you got some competition with Sal.
So, do you have a bad hangover? The worst hangover I ever had was when I was 8 years old. And God bless my parents, they had a certain philosophy that one way they'll teach Sal not to drink and smoke is to give him a little bit, and hopefully it'll make him sick and he'll never. So what happened was there was a part.
I think somebody's got the. Let me just mute you real quick there, Sal. Okay, there we go. Sal, can you? I'm not sure if it was
again?
Sal. I'm not sure if I was the only one, but I could hear a double. Double noise. I can still hear it.
Is it? Are you still hearing me double?
I can hear myself unless it's not. I definitely don't have one in the background. No.
Can
actually, yes.
Am I creating echoes?
John, unmute yourself. Can you hear echo, or is it just me?
I haven't heard echoes, no. Not like we were earlier. I think we're good.
George Bond says sound is good.
Okay. All right.
So, well, this is a good time because I'm just talking about my episode here, the worst hangover I ever had, which is when I was 8 years old. There was my brother-in-law was a bartender at the Officers Club, and he brought this gallon of pink, what I thought was lemonade. And everyone was kind of getting, I guess, tipsy. And I said, can I have a glass of that? And I drank it. So someone gave me a glass. And I said, this is the worst tasting lemonade I ever drank. It was a daiquiri. I didn't know that. I said, oh, what's a daiquiri? So I drank it. The next morning I woke up and I said, Mom, I feel really, really sick. I don't know what's wrong with me. And she said, oh, you have a hangover. So I kind of crawled into elementary school that day, and all my friends were saying, Sal, what's wrong with you? And I said, I got a hangover. And they went to the teacher and told her. And so the teacher asked me, what's wrong with you? And I said, I have a hangover. So I never got into drinking after that. I'll drink wine for communion or something. This does have a point, however, as to the topic of protein probabilities, I assure you. I found other ways to get drunk, unfortunately, not with substance, but with dreams of happily ever after. So I watched movies and I loved happy endings. And when I watched happy movies with happy endings like Star Wars, that just made me feel good. But the reality of a fallen world will start to hit you at some point. There's death, sickness, and destitution. There are ways to deal. There's some coping mechanisms that are a little bit more wholesome than alcohol and drugs, like getting involved, just getting lost in one's career or enjoying sports. It's a little bit more wholesome. But there are tragedies in the world, and especially for Christians, there is persecution. So how do we deal with reality? The Bible teaches, for this momentary light affliction is building for us an eternal weight of glory far beyond all comparison, as we look not to the things that are seen, but to the things that are unseen. For the things that are seen are transient, but the things that are unseen are eternal. And the Apostle Paul wrote, for I consider that the sufferings of this present time are not worth comparing with the glory that is to be revealed in us. And we know that for those who love God, all things work together for good, for those who are called according to his purpose. Who shall separate us from the love of Christ? Shall tribulation or distress or persecution or famine or nakedness or danger or sword? As it is written, for your sake we are being killed all the day long. We are regarded as sheep to be slaughtered. No, in all these things we are more than conquerors through him who loved us. For I am sure that neither death nor life, nor angels, nor rulers, nor things present, nor things to come, nor powers, nor height, nor depth, nor anything else in all creation will be able to separate us from the love of God in Christ Jesus our Lord. And when I was growing up, those were comforting words, but I said, Lord, if this is just belief in something to make you feel good, if it's not true, then it counts for nothing. And so the reason that I began to study creation science and Noah's flood is to gain assurance that this is indeed God's word. And so as we see that the creation account is true, then we have more reason to trust the Bible and what it says, and its promises can be relied upon, certainly more than the promises of man. We do deal with the problem of the invisible and hidden God. It says in Isaiah, surely you are a God who hides himself. And then also in Matthew 11:25, we have this verse that sort of just seems difficult to deal with. It says, I praise you, this is Jesus speaking, I praise you, O Father, Lord of heaven and earth, that you have hidden these things from the wise and the intelligent. There are a variety of reasons that this happens. One of them is that God will disgrace some of the wise and intelligent of the world. That's in 1 Corinthians chapter 1. And then there's also this other verse, one that Kent Hovind cites a lot. For this reason, God sends them a powerful delusion so that they will believe the lie, and so that all will be condemned who have not believed the truth, but have delighted in wickedness. So to some extent, the world is structured, and the activities and events are structured in a way that if someone doesn't love the Lord, God will make it really easy for them to believe lies. So for the topic of tonight, we believe in God through the way he works in our lives. Some of us have been miraculously touched by God's grace through answered prayers, and then through creation and the historic record of Noah's flood. And it says, and so many of us in the 21st century are described by this verse in Peter, though you have not seen him, you love him. And in Romans, the Apostle Paul says, for his invisible attributes, that is God's attributes, namely his eternal power and divine nature, have been clearly perceived ever since the creation of the world in the things that have been made. And that's why we study creation. And Jesus said, if you don't believe my words, believe the works. And some of the works in people's lives are God's miraculous actions. For example, that blind man who saw, he said, I was blind, but now I see. So now that was try to get some motivation for this topic because it is tedious. So I'm trying to give reasons to suffer through some of the tedium. I will try to make it as interesting as possible, but parts of it may be dry. A little bit about me. That is me on the left in Las Vegas, Luxor Casino. Behind me, I'm Salvador Cordova, and I was already introduced, but I'm just repeating my name. And I'm just a random internet troublemaker interested in biology. I was previously a scientist and engineer in the aerospace and defense industry, and then got involved in biology. I'll tell you who real scientists are: Rob Stadler and John Sanford. They are real scientists. I'm just a data clerk by comparison to these gentlemen. So now a little bit off to our topic. Because I was in aviation, I was fascinated. I was involved in the study of navigation. Navigation, to navigate the seas and to navigate around the globe, and this does relate to protein probabilities, and you'll see why, could navigate around the globe. It took thousands of years to develop the maps and techniques. And this was not a trivial task. Lots of people died. We needed very good compasses and clocks and a variety of other instruments, and then building many, many, many charts. So pre-GPS, we had celestial navigation. This is one of the instruments used. It involved maps, clocks, and this device called a sextant. But by the way, some of these slides were part of a presentation I gave at the NIH class, one of my NIH classes in cellular biology. And you'll see how this all relates to cellular biology too. Modern day navigation in an airplane involves a suite of instruments like this. You could see I don't know if you could see my pointer here. That's here's the pilot seat. This is the co-pilot seat. This is the navigator's seat in an old, this is obviously an older kind of airplane. You could see all the instruments needed to do navigation. What's amazing is we have a bird like this called the Arctic tern. It flies point to point 40,000 miles a year. And this was happening before man-made GPS. How it did it, we're only still beginning to learn the mechanics of it. And here was some physicists describing the process. I believe that's a formula from classical mechanics and the Poisson brackets. Oh my goodness, that was the hardest class I ever took. Don't ask me to try to decode that. That is just brutally miserable. So let me point something out about how a bird, like this bird here, is able to navigate 40,000 miles a year. It navigates from the North Pole to the South and back. It's just amazing. And it has all these waypoints in between. It uses quantum mechanics. It uses nanomolecular and nanomolecular magnetic compass, quantum spin chemistry, and quantum entanglement. And this is from Marcos Eberlin. In 2011, a team of physicists used quantum information theory and the widely accepted radical pair model to analyze recent experimental observations of the avian compass. The team concluded that quantum entanglement in the bird's eye lasts about 100 microseconds, exceeding the durations achieved in the best comparable man-made molecular systems, end quote. By the way, Marcus Eberlin is one of the top chemists on the planet. He became a ID proponent. He had 200 PhD students. Not many professors are that good. And that's from his book, Foresight. I presented this in a cell biology class. I didn't know how they were going to react. I didn't talk about ID. I just talked about magnetic sensing and at the cellular level. And I shared that in a class presentation to my fellow students and to the professor. And one of the students, after my lecture, my presentation of only 5 minutes, she said, "Oh, it's so hard to believe how all this could evolve." And lo and behold, did the Lord talk about birds and how marvelous they are. In fact, he encouraged us to study the bird. Did he know something? It says in Luke 12, 24 through 27, "Consider the ravens. They neither sow nor reap. They have neither storehouse nor barn, and yet feeds them. And how much more value are you than the birds? And which of you, by being anxious, can add a single hour to his lifespan? If then you are not able to do as small a thing as that, why are you anxious about the rest?" And he pointed to the creation of a bird. And it's now in the 21st century, this verse has a very different meaning. So now let's move on. Oh, by the way, this protein that is involved in it that uses quantum information theory, it's called the cryptochrome protein. We cannot make a protein from scratch that can do that. If we didn't have the God-made model, we wouldn't be able to build it from scratch. It is that sophisticated. It exceeds anything that we're able to do. And I believe that was deliberate to humble us. So we talk about protein probabilities. It's like, not only can it not be done by random, you take the best scientists in the world, we're not going to be able to rebuild it from scratch if we didn't have a model. That's why I don't believe that many proteins evolve randomly or even by natural selection. We can't build a genetic algorithm to try to calculate that and build that. So let me show another thing. This is the photosynthesis pathway and Calvin cycle in the plant, in a part of the plant called the chloroplast. It is just phenomenal. We cannot build this either. And having I'd taken before I studied biology, I actually my focus was physics, applied physics. And when I was taking a biology, specifically a biochemistry class, I ran across this diagram in my textbook, Leninger Principles of Biochemistry. And it shows here part of the photosystem 1, which is part of the photosynthesis pathway. And you'll see this little photon coming in here, and it creates this thing called an exciton. And I was like, oh my goodness. An exciton is a quantum mechanical quasi-particle. We have to build special solid state chips to be able to implement that. And yet there it is inside a plant. And again, I would challenge, I've talked to biochemists. I said, if you didn't have the blueprints from existing life, would you be able to build something like that from scratch? I haven't met one that said yes. We just don't have the technology. What God has put inside the cell is beyond all of our abilities collectively. In fact, from Scientific American, it says natural photosynthesis turns CO2 into sugars with lots of carbon-carbon bonds, says chemist Andrew Bocarsley of Princeton University, who was not involved with the analysis. We've been studying CO2 chemistry for a long time, more than 100 years, and there's very little evidence that we could do what a leaf does. Of course, plants also have another significant advantage. A bad photosynthetic cell can repair itself. In fact, that's part of its normal operation. No artificial system yet devised, super efficient or otherwise, like solar cells, can heal itself. We don't have self-healing solar cells. So why did I raise the issue of plants? Jesus knew about this. What did he say? So if we take all the wealth of the world, all the technology, all the engineers and scientists, we cannot build something as simple as a leaf. So what did Jesus say to us 2,000 years ago? He said, consider the lilies, how they grow. They neither toil nor spin. Yet I tell you, even Solomon in all his glory was not arrayed like one of these. So does that passage mean a lot more today than probably any time in history? We live in a very special time, and I think God anticipated what we would discover today to inspire us to see these verses in a different light and to know that he's there and he's real and his word is being confirmed. And continuing with this passage, but if God so clothes the grass, which is alive in the field today and tomorrow is thrown into the oven, how much more will he clothe you, O you of little faith? And do not seek what you are to eat and what you are to drink, nor be worried. For all the nations of the world seek after these things, and your Father knows that you need them. Instead, seek his kingdom, and these things will be added to you. So now going back to this article here, it says, natural photosynthesis turns CO2 into sugars. And then it says, it can't, just skipping ahead since I've already read it, it says, no artificial system yet devised, super efficient or otherwise, can heal itself. So I'm going to just pause here. And if you had any reflections, those systems I described need lots of different proteins. You can't build it with just one protein.
No, I'm really enjoying this so far, Sal. Listening to you talk about what God has put inside the cell and how it's greater than anything humans have ever designed, as you pointed out. It really is science fiction on the molecular level. I mean, when I listen to you, Sal, speak on these incredible designs of our amazing creator, it always reminds me of how good these evolutionists are at what? Hoping, dreaming, and imagining that all of this occurred by chance for no reason at all. So, and I really like you giving us some scripture at the beginning, because it's so true. You know, they will be sent a delusion if they want to reject our amazing creator. And no, well said. I'm loving this. John, what are your thoughts, brother?
No, I would second what you just said there. And I think for anybody who, you know, maybe glossing over the significance of what Sal is saying, and the reason I say glossing over is I know from personal experience, I know all those, you know, Sal and SFT, we all have had this discussion with evolutionists about this exact topic in relation to photosynthesis. And when questioned, they gloss over and act like they've explained it somehow, and that it's a totally understandable process that resulted in, you know, photosynthesis, when we don't even understand all of the components of photosynthesis, yet they are claiming with great confidence that they know how it happened, and evolution proves it beyond doubt. And I find that very ironic. One of the things is just a sidebar, and we'll turn back over to Sal, that got me very interested in, much more interested in biology, was when I learned about quantum biology, which I think greatly intrigued Sal as well. And the whole aspect of photosynthesis was actually one of the foundational discoveries that kind of forced quantum physicists to accept that those excitons that Sal was talking about, and how if they're operating in the, you know, coherent state to transfer energy with basically no loss, extraordinarily minimal loss, was one of the things that forced the quantum physicists to recognize that, wow, even though we're unable to pull this off in our systems at room temperature, turns out somehow there is a way to do this. And it really accelerated a lot of the transition into quantum biology, and just all the physicists kind of diving into the research field of biology. I won't get too long-winded there, but Sal, would you agree with what I was saying there, in what kind of kickstarted a lot of the discoveries we've made just in the last decade?
Yes, I agree. So I'm going to go share my screen and
Actually, I'm gonna real quick. George Bond, $5 super chat says, "Sorry, Sal, but this is a well orchestrated trap, and I'm not falling for it. Read a book on evolution. Better yet, Wikipedia." and yeah, that's
Hey,
right,
George, stop plagiarizing Ron Garrett.
Isn't it? It's so funny, the double standard, eh, John? Like, if we were in a debate and our answer to one of the evolutionist questions was "go check Wikipedia," we'd never live it down. But for the evolutionists, they're allowed to say that all day long, apparently. And Doki Doki Bible Club $5 super sticker. Thank you so much. And Honesty Angel says, "Don't lie, SFT. It all goes to pizza." Honesty Angel is very honest. She's on to me. So anyways, thank you so much, guys. And Sal, the floor is yours, brother. Great presentation so far.
thank you. So I was talking about cells that can heal itself. I showed earlier that I work for the MITRE Corporation, which is a congressionally funded think tank. It has a huge budget, like say $500 million a year. I worked in the aviation group, but I briefly worked in their nanomolecular systems group. And one of the two big outstanding problems that they had is being able to create machines that can self-assemble and self-heal. With nanomolecular machines, there's a lot of thermal agitations and the systems are very brittle. They don't last very long. They said, can we figure out how to self-heal? It's still an unsolved problem. So they said, well, one guy stood up and said, oh, we need to study evolution to figure out how it did this. And there were some others in the group who privately were telling me they're just shaking their heads. And that guy would just, he tried to provide his report after just studying the problem over the summer. And I said, did he figure anything out? And they said, no, of course not. There's nothing to learn in evolution to teach you how to build stuff like that. It is advanced technology. So where is that going? Where I was headed with that is biology is somewhat like a 3D printer, but more sophisticated. So here we have a 3D printer. There are printers like this that you could buy on eBay that they're offering it for about $500,000. Now the 3D printer can't print real girls, however. It can't duplicate them, only their contours as pictured here. A cell is a 3D printer that can actually make copies of itself. The formalism of this was described but not worked out in detail by John von Neumann, one of the pioneers of computation. And here's a picture of a 3D printer. You can kind of see here that this printer is making a copy of itself. So it needs to store instructions somewhere, and it has to get little parts and then use those parts to build it. We can't build anything like this unless we pre-manufacture all the parts. We don't have anything yet where we can just build a robot that can take raw materials, process them, and build copies of itself. That technology exceeds anything we can do. And that is the problem with the origin of life. It's not just a mere replicator. It is a replicator that is akin to a von Neumann universal constructor self-replicating automaton. This is non-trivial. This is advanced computer science, advanced engineering, advanced biochemistry, advanced physics, things like this on so many levels. It just kind of makes abiogenesis unbelievable for me. I sooner believe in a creator God and Noah's flood than any of the stories I've heard in abiogenesis. So reviewing back what we talked about in part 1, I talked briefly about collagen, and this is the spelling of the collagen protein, the human alpha-1 paralog. And I think as I showed, I'm just zooming in on it, I showed there is a very noticeable pattern. Not every protein, this is definitely an exception where you can actually see a kind of a syntactical pattern right in the structure of the protein. And I pointed out it's more than just being able to create a pretty syntactical pattern. It requires, for collagen to work, we need at least 3 other proteins, at least 3 others to make it work. They're probably going to be more than that, almost definitely more than that. So the problem is when we say how probable is it to create a functional protein, it's like, well, you need to factor in the probability of all the other proteins that are needed to work with it simultaneously. And so some of the figures by Doug Axe for proteins that function in isolation were actually understated. Doug Axe published this paper in 2004. So the probabilities get more pronounced when we deal with real protein systems rather than the trivial ones. So I covered that. So now what I'm going to point out just a little tutorial here. When we see the spelling of a protein, we call that the primary structure. The primary structure. So there's what they call primary structure, secondary structure, tertiary structure, quaternary structure. So the primary structure is basically the protein sequence itself. It's pretty easy. The tertiary structure is the actual fold. So let me see if I have I don't have a picture of an isolated collagen fold. But just suffice to say, that's the fold of the entire protein. The correct term is polypeptide, or one of its, sometimes we use the word tertiary for the domains, but we don't need to go there. The important thing is we have this thing called the primary structure. And I introduced that to contrast it with what we'll call the quaternary structure. So before I get there, let me pause, because I'm going to show a video on cellular, on DNA replication, and we're just going to have to get through some of the tedious details of DNA replication so we can understand some of the names of the proteins and their functional roles, and then we'll work out some of the probabilities. John, did you have a question?
Well, as you're pulling up that video, I think the point you made about the probabilities and of just for the individual primary structure, as well as the probability need to be incorporated for the dependent other proteins that are required for the function to be executed. And I know from my abiogenesis debates and the books that I've read and the lectures that I've listened to from those in support of abiogenesis, that whole factor is something they do not want to address at all. I mean, in fact, they try to downplay the improbability of just the individual primary structure being created, let alone any of the other subpoints that we're talking about. It really is an interesting fundamental requirement that they try to just act like it doesn't really exist. And I think for those in the audience who are getting ready to observe and look at this video and see the different pieces just for things like DNA transcription, keep in mind what I just said in relation to what Sal is going to be talking about for the probability requirements. These things are recognized as there is no way around them. Would you agree with that, Sal, in terms of even the atheists and the most staunch origin of life researchers all agree that these are things that you can't escape from a how does life come into existence perspective?
They have to account for it. They'll pretend that they're solving the problem, and I'm going to argue that they're not. And they've misrepresented. They've been so desperate they've redefined what the origin of life problem is, so they can pretend that they've solved it. They pretend that they've solved it by creating a RNA replication system, and that's, they're solving a different problem than the one that's there. It's a bait and switch. It's deceptive. It's equivocating, and it's misleading. So I will go kind of into more of the mechanics of seeing what the real problem is. They're solving a different problem and pretending that they're solving the real problem. And I'm going to start trying to define what some of the real problem is. You'll see some components of the problem, and this is just DNA replication. We're not even talking about replicating the cell. And Dr. Change Laura Tan said, DNA is the easiest thing to duplicate. The other parts of the cell, the state that they're in, the glycome, those are much more difficult to replicate. We can copy DNA. We have a very hard time copying a cell in its cellular state, particularly eukaryotes. So I'm going to just start the animation, and I encourage people to pay attention to the helicase and its role, but this is also very good in understanding how we need many simultaneous interacting well-matched parts just to be able to create DNA replication. Without replicating DNA, we can't, cells can't give birth to other cells. So I mean, in a sense, it's dead on arrival in as much as it can't reproduce.
DNA replication is the process by which a duplicate copy of the genetic material is made before a cell divides. The steps in replication are highly conserved in all organisms. There are a few details that differ between bacteria and eukaryotes, but we focus on bacterial replication in this animation for simplicity. The process of DNA replication is broadly described as having 3 phases: initiation, elongation, and termination. Let's begin by considering the initiation phase. For copying to take place, the double helical structure of the DNA must first be opened at a site termed the origin of replication to allow access by the machinery that will copy the DNA. This opening is regulated by a series of initiation regulatory proteins not shown here. Once open, a DNA helicase, a ring of 6 subunits, is loaded onto one strand of the DNA at each side of the replication bubble. This establishes 2 replication forks that will move away from each other as replication proceeds. The synthesis of DNA is catalyzed by an enzyme called DNA polymerase. However, DNA polymerases cannot start a new strand from scratch. Instead, they can only elongate an existing polymer of DNA or RNA. Consequently, a primer sequence needs to be made from which DNA polymerase can continue its synthesis. Primers are made by an enzyme called primase. Once access to the single strands of the DNA has been obtained, the primase is loaded onto the DNA. The primase synthesizes a small stretch of RNA by copying the sequence of nucleotides on the template strand. A DNA polymerase will later elongate this small RNA stretch. The next steps allow loading of the DNA polymerases onto the DNA to copy the entire chromosome. The first component that is loaded is the sliding clamp, a ring-shaped structure that binds at the 3' end of the newly made RNA primer. To get onto DNA and encircle it, the clamp is loaded by a clamp loader complex. The 5-protein clamp loader complex binds to the sliding clamp, and binding of ATP allows opening of the sliding clamp ring. The opened ring then encircles the template primer junction at the 3' end of the primer. Interaction with the 3' end of the DNA stimulates ATP hydrolysis, and the clamp loader dissociates from the DNA to be replaced by the replicative DNA polymerase. The 3' end of the primer is positioned in the polymerase active site to allow addition of nucleotides to this end. During elongation, the replication bubble gets bigger as the helicases move apart, establishing 2 replication forks that move in opposite directions. The sliding clamp and polymerase unit at each replication fork moves along the DNA as the helicase unwinds the double-stranded DNA to expose single strands. Let's focus on just the right-hand fork for the moment, as the processes at both forks are identical. Polymerases are loaded onto both the top and the bottom DNA strands at the fork. Because polymerases only synthesize DNA in the 5' to 3' direction, the 2 strands must be copied in opposite directions. The bottom strand in this fork is the leading strand. The polymerase on this strand moves continuously from left to right, synthesizing DNA as it travels. The top strand is the lagging strand, on which just a short stretch of DNA is made by the polymerase elongating in the 5' to 3' direction. After synthesizing this short stretch of DNA, the polymerase then dissociates and a new polymerase binds at the fork to elongate the next RNA primer. So priming and elongation are happening repeatedly on the lagging strand as the replication fork moves along the DNA. Synthesis is discontinuous. The short stretches of DNA that are made on the lagging strand are called Okazaki fragments after their discoverer. During the course of replication, these fragments are stitched together. When the lagging strand polymerase runs up against a previously made Okazaki fragment while in the process of synthesizing DNA, the replicative DNA polymerase 3 is replaced by DNA polymerase 1, which has 5' to 3' exonuclease activity. This exonuclease then degrades the RNA primer in front of it, synthesizing DNA as it goes. Once the RNA primer is removed, the two sections of DNA are joined by DNA ligase. Now the lagging strand has been elongated by one more Okazaki fragment. While this view of the replication fork is easiest to understand, in reality the leading and lagging polymerases are coupled and move in concert with the helicase. This coupling requires one DNA strand to be looped around so that the lagging polymerase can synthesize DNA in the 5' to 3' direction while moving with the fork. We explore this coupling in more detail in a separate animation. Much less is known about the details of the termination of DNA replication. Once the entire chromosome is copied, the two replication forks meet each other and are dismantled. The ends of the new DNA strands are then joined, again by DNA ligase. The two daughter DNA molecules now consist of one parent strand of DNA shown in grey and one newly made strand shown in green. Thus, DNA replication is termed semi-conservative.
Jungle Jargon said that he'll have to watch that video several times. Actually, that is typical in biochemistry. I've often said biochemistry is like memorizing a phone book, but it's not very different from anyone who has, assembles mechanical devices. You learn the names of all the parts. Like in a car, you have carburetors, you have distributors, batteries, etc., and you connect one thing to another. It's kind of the same mindset in learning the parts. It's just sometimes repetition and kind of connecting it in your mind. When I did biochemistry, the teacher said the way you learn this is you write index cards, and you just kind of recite what you would try to learn, because a lot of it's just like memorizing a phone book. But this is how we see the wonder of what the creator has constructed. It's very much like, in fact, biochemistry books use the word Rube Goldberg machine. In fact, a Nobel laureate used the word Rube Goldberg machine, and that is from, that echoes what Michael Behe said in his book, Darwin's Black Box. The concept of irreducible complexity was introduced in Darwin's Black Box, but a lot of people don't realize Michael Behe also likened biology to a Rube Goldberg machine. And I think that's actually a better description that resonates on so many levels, because biology is way more complicated than it needs to be. It has what people pre-Darwin, they said it had extravagance. It was built in a way not for survival, but to make men wonder at their creator. It had extravagance to it, and I enjoy studying the extravagance more than just arguing with evolutionists. So any comments? And then I'm going to show the helicase. So if you got anything in that video, just remember the early part, we had the helicase, and I'm going to show something about the helicase. So any thoughts, gentlemen?
The only thing I would bring in would be, and it was briefly mentioned, but the ligase actions that are happening. We don't need to get into it right now, but I would strongly recommend people go and Google ligase. Like, the things that it's doing are rather fascinating as well. I mean, just, the helicase and the polymerase, all these things, each one of them, as Sal was saying, they have fascinating functions. And it's very easy for us to get caught up in the more commonly discussed ones. But I recommend folks, if you're a nerd like me, go Google ligase, and you will be yet again blown away by what it does.
All right, I'm going to share my PowerPoint briefly, and then I'm going to share yet another animation. See, I like the format of this. One of the challenges with a debate is you can't get into details like this, and the other side can just bluff and misrepresent, and you just waste all your time not actually dealing with substance. So I'd like to thank SFT for hosting this lecture so that sorry, I got distracted by the chat. SFT or John, I don't follow it carefully. If there's anything you want me to address in the meantime for the audience, I'll try to do that.
No problems. Yeah, no, I'm just going through all the comments, and occasionally I'm putting it up on the screen because our audience is making some really good comments and just loving this. So as always, Brother Sal, we love when you're here. We all learn so much, and I'm loving the visuals. This is definitely another one to remember. So if any questions come up or anything like that, Sal, mostly just comments. Any questions come up, I'll let you know.
Thank you. So I encourage the viewers to look at the helicase in that animation. Now, one thing that they omitted from that animation was topoisomerase, and they did omit a lot of enzymes because obviously they're trying to make it a little bit easier to learn. I have another video which shows the helicase and the topoisomerase. But I'm going to show the spelling of the helicase protein. It's amazing. It's about 1,000, 1,200. In this case, it's this particular helicase in humans is about 1,000. Humans have like 67 helicases, different kinds, just amazing. And so this is one of the simpler helicases, and this is the amino acid spelling. Now, if you look at this, you're like, oh, yeah, big deal. But when you see what it does, that's what will blow us away. So now just remember, this is the sequence for one polypeptide of helicase. And please interrupt me if I confuse you. A functioning helicase needs 6 copies of itself. So to make a functioning helicase, you make 6 copies of this, and then you put the 6 copies together like this. So every colored polypeptide is one of those, the spelling out of those 1,000 amino acids. These are 6 identical copies of each other. So we have, again, this is the primary structure of helicase. Anytime that a functioning complex needs more than one polypeptide to make it work, we say that the complex has quaternary structure. The precise thing is to say it's multimeric quaternary structure. So the helicase has called hexameric, meaning hexa being 6, hexameric, homohexameric, meaning that these are identical 6 copies. So we say that this helicase has homohexameric quaternary structure. Again, I'm just trying to acquaint the viewers that when they hear some of these terms, that's all it really means. So just don't get intimidated by the terminology. It's not that hard to learn. So now I'm gonna unshare this screen, and I'm gonna show the helicase animation. So thank you to the audience for forbearing. StreamYard is a little clunky with shifting between animations, and it's very difficult to embed them in the PowerPoint and then have StreamYard use it. So that's just the way it works. So apologies in advance for that. And if you can maximize the screen, and we'll show the helicase.
Nucleic acid duplexes into their component strands using energy from ATP hydrolysis. The crystal structure of this DNA helicase from bacteriophage T7 reveals a hexagonal arrangement of 6 identical subunits. Surprisingly, the ring is not six-fold symmetric, but is slightly squished. A model for the mechanism of how the enzyme might work explains this structural asymmetry. Of the 6 potential ATP binding sites, 2 opposing ones bind ATP tightly, 2 are more likely to bind ADP and phosphate, and 2 are empty. These 3 states may interconvert in a coordinate fashion as ATP is hydrolyzed, creating a ripple effect that continuously runs around the ring. Because of these conformational changes, the loops that extend into the center hole of the ring that are proposed to bind DNA oscillate up and down as seen in this cross-section. The oscillating loops might pull a DNA strand through the central hole, thus unwinding the double helix in the process. A frontal view shows the full dynamics of this fascinating protein machine.
That machine can rotate in thousands of RPM. I think I heard like 10,000 RPM. And now I'm going to share my PowerPoint. And again, thank you to the viewers for bearing with me having to switch between one and the other. So 10,000 RPM. And then again, what I challenge, not challenge, but I just pose the question, and some of them to biochemists, many of them who are ID friendly. And what I do is I say, can you build something like that? Can you spell the letters if you didn't have a model? Now I'll show you, this is a very simple probability calculation. So there are 2 major phases in naturalistic origins theory. We have abiogenesis, followed then by the phase of evolution. The distinct change when we go to the evolutionary phase is we have a genome. In abiogenesis, I kind of demarcated by saying that abiogenesis is finally achieved when you have a working genome. So what is the problem here? As pointed out, to have a working helicase, you need 6 copies of it. And there's a reason that it's desirable to have 6 copies. If you're going to wrap around something, like here's a nut, you want to have a bolt that goes through here, you have to have symmetry. So it totally makes sense to have something that's symmetric. So again, you need 6 copies of this or something, or some facsimile of that. So this leads to a probability calculation. What are the odds that you can repeat this pattern 6 times identically? This is almost 1,000 amino acids. That would be something on the order of 10 to the 6,000th power. And some evolutionists like CRISPR, he'll say, well, it's only 50% conserved. I said, okay, fine. Let's make it, that'll make it 10 to the, on the order of 10 to the 3,000th power. And anything higher than 10 to the 150th power, the practical limit of believability in the observable universe. So this is a very simple calculation in an abiogenesis scenario. So how do you,
quick question,
go on.
Does that that calculation you're talking about there, is that just calculating the amino acid very possible variables, or is that even accounting for the requirements in terms of the actual sequence, like the codon variables? That's just saying, hey, if all the amino acids just did it by themselves, we don't have to, let's not even worry about the actual sequence having to also account for it. Let's just look at it from a raw spelling perspective. Is that what you're saying?
That's exactly what I'm saying. It's just like playing a game of poker and the dealer deals out the same 52 cards.
So in the context of
six times in a row, he'll do it 6 times in a row. And actually, one time a dealer got caught doing that. The FBI watched him. They realized, oh my goodness, he's doing false shuffles, and they threw these guys in jail. But they're a really good team.
So a quick expansion on that. So correct me if I'm wrong, but as far as I know, the Origin of Life community has pretty much abandoned cell formation of polypeptides, right?
I have no idea, but here's one of the problems. One way that people say, well, that problem's obviously solved. Once you have a genome, you can easily make polypeptides that are identical because now you have the recipe and can repeat it. But as you could see with DNA replication, you need something like Helicase to make a genome functional to begin with.
And that's why I was asking, is what you were talking about from an improbability standpoint even bothering to account for the formation of the genome sequence? That's the amazing portion of all this. I know we'll let you get back to it, but that's one thing I think everybody needs to really remember because just in terms of getting the sequence, you got to be sitting there doing however many nucleotides to the 4th power, or that number to the 4th power, and then you have to calculate it. The math becomes completely. It goes beyond what humans are capable of truly comprehending, and I think that's not just me saying that as a Christian and somebody who believes in creation. That's a recognized premise that the human mind is incapable of comprehending the true level of fantastical probabilities that are being discussed.
Yes. So this is a very simple probability analysis. And only this year, someone by the name of Thornton finally fessed up to the problem of multimeric proteins. He claimed to have solved it through phylogeny, which is baloney. He actually said, he tried to make it seem that he solved the problem, but even he had to admit it's fortuitous. He said, the problem of multimeric proteins is outstanding. I'm just like, yeah, you guys never talk about it because it's embarrassing. It's so bad you don't even want to acknowledge it. And this is a multimeric protein. This is a protein with quaternary structure, multimeric. Multimeric meaning just basically you have multiple polypeptides, which are basically these colored blobs here, which are the polypeptides. And I gave the spelling of each of those blobs. They're identical. There's the spelling of it. So you just repeat that. This is like a dealer dealing out the same sequence of cards. But in this case, this is 1,000. This is 1,000. Just try doing that. This is very simple calculation.
So one more question for you. So right now we're talking about the same quaternary requirements, and you use the analogy of getting the same hand. You got a kings over queens full house 6 times in a row. If something is hetero versus homo, now you're talking about not only do you have to get specific hands, you have to get. This one has to be a jack high heart flush, straight flush. Yep. The next one's got to be a ace high flush. The next one's got to be a full house, and then so on and so forth. Is that a fair, logical comparison?
Yes, and of course they'll say, well, the pre-existing one didn't have to be that way, and I'm like, well, that's why I pointed out symmetry is actually really important in machines. Like if you're going to have a rotating part, it kind of makes sense to have it symmetric. So there's actually a physical, mechanical reason we want to have it repeating, just like this repeating nut here. You could kind of almost see it there. They're mechanical considerations that you need that many repeats. So that's just not an arbitrary thing that I threw in there. It totally makes sense. That's why we have lots of homomeric quaternary structures where you see all this symmetry. You have things that look like scissors. You have things that look like nuts, or anything that are where you need symmetry. It's just all over the place. By the way, this is kind of like a hexagon, right? 6 parts, and definitely the nut is a hexagon here, and the hexagon is optimal. Like in a honeycomb, there's actually a mathematical theorem called the honeycomb conjecture, honeycomb theorem, hexagons are optimal in certain contexts. So I didn't make that up. This is why I'm just like, guys, ID community, there's some things that are practically being handed to you. You just need to look at the probability calculations just a little different way, and you'll kill the other side. They're not going to be able to contest the fact that you need a genome to be able to do some of these things, because there's lots of homomeric and then, as you say, heteromeric proteins. I mean, they won't work without multiple copies. So you need a genome to make a genome, and that's a chicken and the egg paradox. And you know, some people say, well, you don't need exactly that many. I'm like, okay, well then tell me, okay, let's go with 50%. You still have you just have a combinatorial disaster if one is invoking naturalistic means. And I haven't heard this argument, which I just gave you, successfully refuted. And finally, Thornton, an evolutionary, he's actually a chemist who's kind of pro-evolution. He admitted that this is a big problem, not just for origin of life, but for evolution in general. He studied the hemoglobin heteromeric complexes, by the way. So I'm glad you totally get this, especially since you and I have played cards.
A quick interjection. I know we don't have tons of time left, so I don't want to distract you too much. But something the audience should Google if they don't think what Sal was talking about in terms of the hexagonal, the importance of that structure. If you go and Google it, you will find that there are skyscrapers and there are modern buildings that architects are now beginning to have the capacity from a construction perspective to incorporate that into their architectural structure. And they're trying to pull this off because they now realize this for stability and space and there's all these different factors that are relevant in context of this. We're now trying to incorporate this into our own buildings. And so if anybody says that doesn't have relevance from an engineering and architectural perspective, they're just ignorant and wrong. And they should go Google it for themselves.
Thank you. And by the way, this is just one of the critical proteins in the replication cycle. We're not even talking about translation and transcription or anything else, just this one. So just for the readers, for the viewers' benefit, SFT said we can host more of these because I said, SFT, we probably need part 3, part 4, part 5. So this is part 2, and I don't know how long this series is going to go.
I'm happy to make it go all the way up to part 100, Sal, because these are, and one thing I love about these lectures too, not only do I learn so much, they are so rewatchable. And many people in our audience agree with that. You know, once the lecture's done, it's beneficial and it's fun to go rewatch it, and you learn something new every time. So Sal, we can do 100 of these. These are awesome.
Oh, thank you. So one thing that abiogenesis proponents and evolutionists say, oh, you're just you're having this single target probability objection. They call it the Texas sharpshooter fallacy, where you say, oh, well, you know, it's kind of after the fact. That you just said it's improbable because you can't get that same thing again. Well,
you shot a wall and then you went and drew the target around it and said, wow, look at this bullseye.
Right. But the problem is, that's why I like this argument with the homomeric proteins. It kind of blows that away. It's like, well, it doesn't matter what the probability argument is not saying we need this specific sequence once. The probability argument that I'm putting forward at this stage, there are many others I could put forward, is the difficulty is making the same sequence by random chance 6 times over. And so that will circumvent this, that circumvents this single target objection, the Texas sharpshooter fallacy. It falls apart, gone. They can't make that argument with this. And there are other ways to approach this, but this is a very easy one. And Now, when Erica showed up at the after show, she was saying, "Oh, you only need you have the world, and it has trillions and trillions of molecules and atoms. And one calculation that I saw is 10 to the 50th." And so she's suggesting that there's so many trials on the planet, you're bound to eventually get a cell. Well, Seth Lloyd, I believe he was from MIT, he did a calculation. And it is likened to the probability of getting fair coins tossed. If you had 500 fair coins and you tossed them and they all ended up heads, the odds of that are so remote, you wouldn't expect it to happen in the observable universe. Even if you had the resources of all the possible quantum interactions, anything that needed a probability of 2 to the 500th power, 1 in 2 to the 500th power, it's just not going to happen, practically speaking. You wouldn't even remotely want to build a scientific theory or make a claim and represent it as scientific. You can make the claim that chance can do it, but it doesn't accord with what is the expected outcome of physics and chemistry and probability. It would be like Jim Carrey saying, "Yes, I have a chance." So I don't, Erica Gutsick Gibbons cited that, and that's easily overcome. If you can create probabilities that exceed this universal probability bound, practically speaking, it's outside of scientific verification. It's just built on faith. It's not an inference that you should be making and claiming it's scientific. Now, what about the origin of life community? Whenever I make all these arguments, they'll just say, "Oh, well, look at all these 13,000 papers that are being published on the origin of life." I liken to what their work is like, "Oh, we're showing the early steps of Humpty Dumpty about to fall out of the wall, and we're going to show he's not going to break when he hits the ground, when he hits the rock hard ground." This is what the origin of life community and researchers are doing. They are pretending that they're going to solve the Humpty Dumpty problem, that Humpty Dumpty, by way of figure of speech, by analogy, they're saying, well, we're on our way to proving that Humpty Dumpty won't break when he hits the ground or when he hits the pavement. Look at all the papers we've done. We're demonstrating it. We're making progress. And that's just an illusion. James Tour called them out on it and said origin of life research is retarded. And he was instrumental in enlightening the Nobel Prize winner, the late Richard Smalley, while he was alive, enlightening him on the origin of life. Richard Smalley won the Nobel Prize in chemistry, and he's called the father of nanotechnology. And praise God, through Jim Tour worked for Richard Smalley. It was through his witness that Richard Smalley became a Christian before he died of cancer. So praise God.
Hey,
and Richard's, yes.
Question. The gentleman you're referring to, was he the same chemist who, I think it was in 84, 85, was it his team that developed the first molecular circle or molecular ring or something? That they were
the buckyball.
Yes.
Yeah, he's famous for the buckyball. Yep, that's him.
Something for that to the audience, I think you should recognize is he basically proved that you can make shapes out of molecules that have a specific objective. And
Amazing.
they won a Nobel Prize. It was just phenomenal achievements, et cetera, et cetera. But I think the reason I'm bringing this up is, as you saw in the animations that Sal's been showing in this lecture and previous, and I think in the future, this gentleman who he's referring to, Smalley, won a Nobel Prize for creating something that is nothing compared to what are fundamental requirements for life to exist. But I would wager, Sal, correct me if you think I'm wrong on this, but I would assume that when Dr. Tour was explaining all of this, what's happening in biochemistry to this Nobel Prize winning chemist, since he knew how hard it was to pull off the things that he won a Nobel Prize for, I would assume it probably resonated with him rather rapidly of just, wait a minute, what are we being told happened? We really started to understand the fundamentals of what's going on.
Yeah, but John, Sal, you guys are forgetting about somebody. As you can see here, Brother Bill Morgan says, godless engineer. He has single-handedly solved the origin of life problem and all of its chicken and egg problems. So we're running from him. That's why Bill says, are you willing to debate the godless engineer, Sal? DNA is like Coke.
Yeah. Well, I'm going to move on. And this is what happens to Humpty Dumpty. It's called the asphalt paradox. And it was written by an origin of life researcher, Steve Benner. He said, the asphalt paradox, an enormous amount of empirical data have established as a rule that organic systems given energy and left to themselves devolve to give uselessly complex mixtures, asphalts, quote unquote. It's not literally our asphalt in the ordinary sense. It's just a figure of speech to talk about uselessly complex mixtures. Theory that enumerates small molecule space as well as structure theory and chemistry can be construed to regard this devolution a necessary consequence of theory. And I have a little bit of, this is from Raiders of the Lost Ark. This is kind of how I visualize the asphalt paradox. And there are more paradoxes that he highlights. So if you don't have a functioning helicase, a lot of all that's just gonna devolve by itself. That's the natural direction. And we'll talk about, I'm gonna try to wrap up. It looks like the parts that I had written for today will have to be deferred to part 3, where I really pounce on Jackson Wheat's, one of his statements about leaky membranes. But let me try to wrap this up a little bit. So what does it mean that something happens naturally? If we take the metaphysical notion of natural, if we just talk about, if we use the word natural to mean what is normally expected, normative expectation, just naturally. So if we have, like, say, this is actually an electrical engineering diagram when they talk about stable circuits. But they use the analogy of a bowl and a little marble here. If you drop a marble into the bowl, and because there's a little bit of friction, even if you begin at random locations within the bowl, the marble will eventually settle down to the same final state. That is an important feature of what we mean when something will happen naturally. It's natural then for a marble to eventually, even though it starts out in random or uncertain points within the bowl, assuming we don't give it initial velocity, it's going to end up at the bottom. And there, these other systems, we won't, I should have omitted those others. But that's what we mean by natural. Now, similarly, if we have hydrogen molecules, hydrogen atoms and oxygen atoms in random orientations and positions within a rocket engine, when you mix them and you light them up, they're going to combine to and release energy, but they'll make water molecules. Here's a Saturn V rocket making lots of water molecules. In this case, the first stage of the rocket uses kerosene and oxygen. One of the second stages uses hydrogen and oxygen. You'll get this tremendous amount of fire coming out, but then you just generate lots of water, and you get a predictable structure like that, which is nice. That's what will happen, quote unquote, naturally. And they're defined angles and structures. But there are chemical systems that lack that sort of predictability as to what is the final structure of all the elements, all the atoms. We can illustrate this with, say, Scrabble parts. Now, with cards, if you throw them, like, say, randomly, they'll usually land in this horizontal position. But there are states that are possible, but we don't expect them, like this state where you have the cards in, like, say, a tent. and it's on its way to be building a house of cards. This is not expected from cards being just blown about by, say, a mild tornado or a wind. This would not be surprising, a layout of cards like that. This would be surprising. We would reasonably infer design because we have human designers. But from a probability standpoint, we could say from expectation, this is not what we would expect from random processes. So unlike the hydrogen atom that can start off from very uncertain or a variety of states and you end up with a predictable geometry, we can't do this. We can't, for cards, it will not predict this. And this has analogy to organic chemistry. And that's what Steve Benner, I believe, was, that's the way I interpret what he said. Is structure theory is that it won't evolve into these coordinated structures. You could see the coordination here. And again, like Helicase, if you see there are 3 tenths here that are relatively identical, that's very hard to duplicate by random chance. Not to mention it's a difficult geometry to achieve just by random motions of wind. I posed this problem to Snake Was Right, and Snake Was Right was actually wrong. When I said, don't you think that that's improbable? He said, yeah, sort of. I'm just like, yeah, emphatically, you ought to be more emphatic than that. So in the molecular world, I'll illustrate it with Scrabble pieces. Let's say, independent of the spelling, we just want to get the Scrabble pieces kind of in a position that they can be read. And this is analogous to DNA. So this is not very readable like that. This one, the Scrabble piece, has the letter facing down, quote unquote. So what has to happen is it has to be arranged. And now it's getting close. Now it is almost, now it's readable. This has analogy to DNA, that the molecules involved, the adenine, thymine, etc., in a random chemical soup, it will not organize itself so that you have this kind of like railroad track here. Change Tan, who's a physical organic chemist, showed that you'll get so many conformations. She estimated just with 2 nucleotides that randomly formed in a prebiotic suit, if it's adenine and cytosine, that there's over 16,000 possible ways of connecting it. And she worked that out in her book co-authored with Rob Stadler, and Rob Stadler here appeared on the SFT channel. She details how that calculation is done in her book, The Stairway to Life. So even without getting to the more complex part of the equally or the complex part of the translation system, you need to have the hardware in a point it can even read and write. Because in a prebiotic soup, you don't even have things where they can read and write. They're not in a state that's readable. The DNA, the nucleotides won't be in a state that it can be read by transcription, translation, and replication machinery. So even just talking about the hardware, the basics of the hardware getting straightened out, it's highly improbable. And these are the problems with the origin of life.
And so I think the quick expansion on the point that Dr. Chan was making is to expand on the like the Scrabble analogy. Imagine, folks, that there are all these different little magnets all over each one of the, like all over those, each Scrabble piece. And there's north and south poles. You have to not only get all of the correct letters, you also have to have the magnets have all that are on them have lined up perfectly in order to get the readable word in order. But there's all these other ways that, and the point you're making, it's like 16,000, right? Isn't that what you're saying?
Yeah, over for adenine and cytosine. Yeah,
so there's
just for two nucleotides. We're not even talking about something that's 100 nucleotides.
Right. So you have all of these different possible ways they can bind, just like all these different ways that those magnets could potentially click together, yet there's only two options you can, or sorry, only one pair option you can choose from in order to get the end outcome and have the correct formation at the end objective. And I think that's something that gets glossed over quite aggressively by the origin of life researchers. And I know it's one of the things about structural biology that has you so fascinated on the chemistry level is that's what we're talking about. We're not talking about just purely linear information. We're also talking about the three-dimensional structure that it must be in place in order to derive the linear spelling that we're looking at in down the road.
And this is we're headed toward the end of the discussion. How do cells do this? They need proteins. And I'll tell you how, I'm going to emphasize it again. We tried, when Craig Ventnor claimed that he made artificial life, he was trying to synthesize the genome from scratch. And they had to use this thing that's analogous to what they call the blue heron process, where they had to orient the molecules, the nucleotides, in the right position. They also had to start out with the homochiral. DNA also has its kind of chirality. It had to be the right one, and the process was painstakingly slow. What they had to do is they put an abundance of, say, you needed an adenine. Okay, so let's make everything adenine in the beaker, quote unquote. It's really the assay. It's a very small assay, and so that it's going to force adenine to be added because they're trying to exclude everything else. So once that's added, it's like, okay, now remove the adenine. If you need something else, add that. And they would just keep repeating this process of adding a chemical into the beaker, quote unquote, figuratively speaking, and then emptying the beaker and then adding the next chemical. And you do this slowly, very slowly. In cells, this is just done like lightning speed by comparison. The other thing is, they had to orient the molecules just the right way. So you needed the right species, but then you needed all this machinery to position the molecules. It's analogous to when I was positioning, see like how this is kind of upside down, or I'm just doing this by analogy. They needed a whole bunch of specialized chemicals to orient the nucleotides so that they would connect in the right way before they connected. And I think they could only get 100 nucleotide polymers. It was so bad. Beyond that, they couldn't get anything stable. So then they had to stitch all these 100 nucleotide segments, what they call cassettes, to make a genome that's 500,000 big. It was brutal. Yet the cell does this so effortlessly because it has all the right machinery. Again, we're seeing that the humans look clumsy. They look like they're making paper airplanes compared to the space shuttle, the way that we were going about it. And I just saw Jungle Jargon say, Craig Ventnor tells fibs. He exaggerates, definitely. He didn't create life. God created life. He had to borrow the parts to make it. And even then, as Dr. Tour pointed out, there was just it was more or less transplantation. He wasn't building this from scratch. And DNA, Dr. Tan pointed out, DNA is the easiest macromolecule system to duplicate. The other parts of the system, like the proteins, with their glycosylation and their post-translational modifications and all the other stuff, and the sugar code in the cell, just forget it. We can't duplicate it. We are humbled by what God has put in the cell. The tiniest thing, the very thing that we neglect, that we don't hardly even see or we give thought to, is the very thing where God is showcasing his engineering. So I had more in the presentation, but I'm gonna have to defer it to part 3, and we're gonna revisit the transmembrane proteins because this discussion covered abiogenesis a little bit. The next one, when we do transmembrane proteins, we'll cover the problem of eukaryotic evolution So they'll
Yeah.
say if the critics say, well, you're talking about abiogenesis and it's not evolution, I'll say, okay, well, that's fine. We'll talk evolution next time. We'll talk transmembrane proteins, and we're going to show why Jackson Wheat basically dropped. I'll show you what he did. I'll share
Yeah.
my.
Yes, do it again. We should show it every single lecture, because I think we discussed that last lecture as well. And
So, Standing, you know what's happening right here.
what's happening, brother?
This is what this Sal is so nice. However, all of the little YouTube atheist evolutionists running around just need to recognize that when they issue with such confidence their completely debunked arguments, they will trigger Sal Cordova into creating an entire presentation with the express purpose of pointing out how dumb their arguments are, how they've been debunked, and how if they actually would stop listening to the talking points and maybe do some contemplation for themselves, they would be able to recognize the glory of God and stop relying on things that have been debunked quite some time ago. So go ahead,
Well,
Sal.
it's amazing how many lectures we've now had with Sal on, and I mean, evolutionary fitness, protein probabilities 1 and 2. 6 months or so ago, we had Sal on for a number of must-watch, must-listen-to lectures, and not one real serious rebuttal or counter argument. They really are, as I always say, team dodgeball. And we can safely add Jackson Wheat to the roster of team dodgeball because, Brother Sal, if you want to touch on
I'll
Jackson
touch on it. Okay. I was criticizing eukaryotic evolution. I said there's a problem in forming transmembrane proteins. The Jackson Wheat, he said, oh no, he suggested that wasn't a problem. You just need a little leak in the membrane. And I was nice at the time not because I'm really not that nice a guy, but I'm kind of reserved. It's like, okay, I'm not gonna hammer you on this today. Let me go and read up on this. So that was December 2018. I had since studied more biology in the intervening 2 years and had the chance to work with Change Tan and Joe DeWeese and other professional biologists and biochemists. And I learned stuff. And I knew he was wrong, but I didn't want to call him out on it. He made this is what an evolutionist does. He'll just dismiss the problem and say, he'll just make something up on the fly. And I think this is what other evolutionists have done. They said, oh yeah, the eukaryote can evolve. It just needs these leaky membranes to make stuff go in and out. And I got to call a cow pie. Jackson Wheat is dumping cow pies, and I'm just a janitor trying to clean up all the poop that these guys are generating. That's what a creationist has to do now.
That's a significant amount of bovine scatology right there.
Yeah. So next lecture is the cleanup of Jackson Wheat's cow pies. I mean, man, it just doesn't take a lot to just dump a pile of garbage, and it's just such a mess to clean it up. And I spent 2 years on and off looking at the problem of transmembrane evolution to form eukaryotes. And in the process, my jaw also dropped as I was preparing this material. I said, boy, there's angles of this that people aren't talking about, and it's a real problem. So next week, I'm going to be pouncing on Jackson Wheat's statement and also all the people that are wanting for creationists to go beyond abiogenesis. The next step is eukaryotic evolution. And by the way, this also relates to universal common ancestry. If you'll notice, even Erica was kind of stunned when I pointed out, I said, your colleagues don't believe in universal common ancestry of protein families. She's like, no. She didn't say no literally, but she was like, huh? And
you know something else that's interesting about that, Sal, I think this dovetails what your point is. The expansion point of Douglas Axe and others since him is not just on the formation from a probability perspective. It's not just the original primary structure formation. It's then what is the A to B probability? And I watched an interesting interview with them talking about that whole deal, and it's basically function A going to a completely novel function B, for all intents and purposes, impossible from a probability perspective. Protein A modifying to now execute a pre-existing function in that side of that structure is not. But protein A transferring through evolution, through random process into a completely new function is an entirely different dynamic. And I thought it was very interesting listening to them talk about that. I'm assuming you'll probably dive into that in more detail. But that whole piece of it, in my opinion, completely annihilates the entire premise of evolution, and that universal common ancestry, whether it be just from a phenotype or phylogenetic perspective, as well as just proteins. It's a whole new animal. You're on mute, Sal.
Sorry. This could be a very fun topic, and how it relates to common ancestry of proteins. And it's really funny. The other side has basically admitted it. And I'm like, okay, you've already taken one step into the creationist model of created kinds. I mean, really? I mean, what's stopping you when you can dispense with common ancestry of proteins? What's stopping you from dispensing with common ancestry, period? Really?
Well, I think one of the, I know we got to wrap up here, but I think one of the comments, you've used this similar argument, I use it all the time, of, and I think they're being forced to come to this conclusion, is everything about common ancestry and huge swaths of evolutionary theory in and of itself, they're being forced to come to the realization that it's all fruit of a poison tree. Like, there is no way around this. Like, if you can't account for common ancestry, whether of kinds or proteins, what is evolution then? Like,
Yeah, it's
if there
useless.
is no, if that is flawed, and that is not a remotely plausible explanation, then you got to start again. You got to come up with something else. Either you can accept the fact that we were created, or you better come up with an entirely new paradigm. And I think that's what they're balking at. But some, as you've talked about, are starting to recognize that, wow, there's no way around this. Like, we've got to account for this at this point.
Yeah. So my focus has been on the bottom of the phylogenetic tree. Standing for Truth is focused at the top, quote unquote, the human origins. I've focused on abiogenesis and eukaryotic evolution, which is very much at the bottom of the phylogenetic tree. So I'd like to thank all the listeners and viewers who joined us today. There's going to be an after show. I just need to get my my StreamYard spooled up, and as soon as this stream is done, I look forward to meeting some of you there. And I'm going to try to terminate it for those who are really just really loving this and getting high on information. I'll try to terminate the after show so that people can hop on to Standing for Truth's debunking of Dr. Dan.
Well, and just to make one comment on what you guys were saying, here's the thing: without any explanation for the origin of life, chemical evolution, naturalism is dead in the water. So I love what you're doing, Sal, and I'm really, really pumped for next week. I'm gonna make sure that when we seal the deal on a specific date and time, I'm gonna advertise that event. That'll be part 3. I'll advertise it a few days in advance to get people
thank you.
notified about it, because I'm not the only one who's going to be extremely pumped for that one. So maybe next week we'll do it.
Standing, I think you should pick those ads to run on Jackson's channel and RJ Downard's just for
Yeah, really
just for
love it.
entertainment purposes. Ladies and gentlemen, this Sunday at 9 p.m. Central Time, come on over to Standing for Truth for the epic debunking of the guy on whose channel you're watching right now. Salvador Cordova will be educating Jackson Wheat on what actually constitutes a leaky membrane.
I'm telling you, Sal, you are the new evolutionist nemesis. And we're trying to hopefully get a debate set up between Sal and an evolutionist on evolutionary fitness. So we'll see who's willing. But if I was an evolutionist, I would not want to debate Sal. So, Sal,
has someone accepted?
Well, we're hoping Dapper Dino. You know, you and I, we are coordinating.
He hasn't responded to you yet.
He hasn't. He agreed to the debate. Now he just needs to respond as to the date and time of the debate. So hopefully we get that set. Looking forward to that one. And tonight's lecture, before we end it, I want to say it was awesome. I love the way it flowed. I love the visuals. And it's definitely one I'm personally going to go rewatch. So anybody in the audience who just got here, it's been over an hour and a half, and we've maintained over 30 the whole time with a few new viewers. Make sure you go back at some point and rewatch it, because so much good information in this lecture. Go back, watch part 1 as well. And I just set up a Facebook page for Standing for Truth Creation Ministry. Go like it and follow it, so you can catch all the notifications for these upcoming streams as well. And last thing, we've got an after show on Sal's channel that's going to kick off a few minutes after this one. So make sure you're there. It's going to be a lot of fun. And any last words from you, brothers? All righty.
Praise the Lord Jesus Christ. I acknowledge him. He said to acknowledge him in all our ways. And I'd like to thank the Lord. Something that I've been praying with I have a small private creationist group. And I've said that there's a passage in Ephesians that God has prepared for us works before the foundation of the world. And I think this meeting and discussion is something that the Lord had ordained even before we were born. And it kind of adds nobility to what we're doing here tonight, even though many people don't hear us. This means a lot to the Lord, and I acknowledge him for that. So thank you.
Amen. Amen. I love it, Sal. It's not about winning arguments. It's about winning souls. So we want this information. We want the truth to reach as many people as possible in order that they can hear the gospel and come to salvation. So well said, Sal. This was a wonderful lecture. Fascinating. So thank you again for your time. And John, I'll let you have the last words, brother. Thanks for being here, and thanks for contributing to the talk tonight.
Oh, well, I would agree with everything Sal said, and I'll end with Proverbs 3:6, which is, trust in the Lord with all your heart and lean not unto your own understanding. In all your ways acknowledge him, and he will make your paths straight. Be not wise in your own eyes. Fear the Lord, and turn away from evil.
Amen, brother. Amen. Well said. Well said. Beautiful way to end this. Everybody, be there for the after show, and be there tonight around 2. After the after show, take your time on the after show. I'm a night owl anyways, and we'll have a bonus round debunking Dan. So everybody have a good night, starting with the after show after this, and SFT is out.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are